Well, thank you, Britt, and thanks for the invitation to come here and join you on your Brain Day event today. It is true, my postdoctoral advisor wasn't much of a fan of attention. In fact, after we'd done our experiments and collected a lot of neurophysiological data from monkeys who were doing different attention tasks, we wrote up the article for submission, and he sat me down and said, "This word attention won't do. Nobody knows what attention is. It hasn't got a good proper definition. We're going to call it behavioral state," which we did. I managed to leave the word attention as one of the key words. I don't think he noticed that I'd done that. But he effectively made a purge of the word attention throughout the article. And I have to confess, it didn't actually change the content or the message of the article very much at all. But it left me disappointed because attention is a topic I really wanted to study and have studied since. And I've reverted to using the word attention. But it's been with the acknowledgement that we really still, to this day, don't have a good, comprehensive, agreed-upon definition about what attention is. Every one of you has a pretty clear idea about what I'm talking about. But the fact is, we use the word to describe many different phenomena. So, for example, attention, we often think about some sort of task where we need executive control over an extended period of time, like driving home. We need to do a lot of different things to complete that task. It may take tens of minutes, may take an hour or more, but we can sort of direct attention in ways that allow us to get there successfully, even though sometimes at the end of it all, we may not remember much about what we were doing during that process. At the other extreme, we use attention for attention-grabbing things. If a firecracker goes off behind you, you will orient to it. It will grab your attention in an involuntary sort of way and force you to orient to that location. And in between, we can, over shorter periods, direct attention in a sort of voluntary way to specific stimuli on demand as we like, on instruction or based on our interest on what's going on in the scene. So there's a whole range of phenomena, some that we associate with top-down decisions that we make about what we will attend to, others that are called bottom-up, that are sort of driven by stimuli in the environment, some more directed towards tasks, some more directed towards stimuli. But it's not clear whether they all engage the same mechanisms in the brain. They all involve this sort of notion of sequential processing of events over short intervals, perhaps fractions of seconds, perhaps a second or two, as we go from one step to the next in whatever it is we're doing. We attend to one thing or another, and it's possible that all of these involve that same sort of process in the brain, but we simply don't know. And so my lab's been working on trying to understand more about what intention means in the brain, and I'm going to tell you about experiments we've been doing, recording from individual neurons in the cerebral cortex, primarily, of rhesus monkeys, who've been trained to do tasks that direct their attention towards or away from various stimuli in the environment, and we look to see how their attention affects the way that sensory neurons respond to those stimuli. And in particular, I want to focus on the recent experiments that we've been doing that have been trying to understand to what extent can we subdivide attention. Is attention really just one process, or as sort of implied by these sorts of phenomena, it might actually be subdivided into different categories? And as neurobiologists, we think that there's actually good reason to expect that it might be subdivided into different phenomena. And our model has been memory, which 50 years ago was one thing. We knew that there were cells with connections in the brains that had to change to lay down memories, but there wasn't a great distinction made between different types of memory. And I won't go through all of the different classes that have been worked out here, but I'd say about 20 years ago, it became clear that memory was not a monolithic phenomenon. There are different types of memory, and you can actually subdivide things into at least a crude taxonomy that addresses those different types of memory. And most importantly, from our perspective, is those different types of memory seem to map onto different neurobiological structures in the brain. And each has its own contribution. They work together as a kind of confederation to give you all the types of memory that you need to perform the various functions that you undertake. And it seems unlikely that attention is as richly subdivided as memory is, but it's something that's never really been explored in much detail. And so this was an effort that we wanted to make. And before describing the specific experiments, I have to acknowledge we can't study all of attention. And so we focused on a very narrow part of it, and I want to just make clear how much we've thrown out in the process of coming to the experiments that we do. So of these different types of attention, bottom-up attention is hard for us to study in neurophysiology experiments because it involves setting off a firecracker or some other alerting or orienting stimulus and then watching how attention changes. But in that situation, it's really hard to separate the sensory stimuli from whatever consequences you get in the cognitive allocation of attention. Things change, but is the change you see related to the stimulus or is it related to attention? It just gets very difficult to sort things out. So we don't study that kind of bottom-up attention. We also don't study executive control because it's also difficult to know what's going on there. Executive control is an organism sort of deciding to do a sequence of tasks in an order to reach a goal. It's up to them what they're doing. We don't know what's actually going on in terms of what's the local sequence of processing that's going to occur. So as a neurophysiologist watching what a neuron's doing, we can see changes, but we really don't know what to relate them to in terms of the control. And so there's this happy middle ground where we think we can get some leverage and begin to understand how activity of cells varies as you change attention towards or away from specific stimuli over a period of a few seconds or so. And this is something you do all the time. The sort of classic example is when you're in a room full of people all talking to each other and you sort of focus in on one conversation behind you to the exclusion of all others. And you can switch between them, right? This is what you do. You basically select one set of auditory input to process and select others for filtering out. And by doing that, you can actually get more information for your current purposes. We don't study the auditory system, we study the visual system because of several good reasons. The main reason is that it's the best studied sensory system. We have more foundation in terms of what neurons do and how the brain is organized to study the visual system than we do for other sensory systems. And these are just maps of the rhesus monkey's brain's cerebral cortex, sort of stylized here a little bit, but the different colored areas represent discrete functional units, different areas of the cerebral cortex that are devoted to processing different types of signals. The visual part of the monkey's brain is all of this part, what would be the back half of the brain. It's approaching a third or a half of the monkey's brain devoted to vision. It's a little surprising. It's more than all the other sensory modalities made up together. But in fact, it's appropriate. Primates are extremely visually oriented animals, and so they have more computational power devoted to vision. We are primates, so we're really interested in vision, and there's been a lot of studies that have looked at how the visual cortex is organized, both in monkeys and in humans, and I'd say the organization is quite similar across all primates. Each of these areas represents a different visual area. V1, the first area here, which is at the back of the brain, is the first visual area. The signals coming from the retina arrive first in V1, processed there, and then are passed out to subsequent areas in later stages of visual cortex, which number about 30 or so in the rhesus monkeys. Each of these areas, we think, has its own representation of the visual scene and is specialized for representing a different type of information. So MT, the middle temporal visual area, virtually exclusively devoted to representing directions of motion. Those cells are really exquisitely sensitive to whether a stimulus is moving in a particular direction or a slightly different direction. But they're insensitive to properties like color, size, shape, form, contrast, texture. They really don't care what visual stimulus is moving. They just signal the movement that's occurring in the part of the visual field where their receptive field lies. Next door, V4, V4, an area that I'll be talking about today, doesn't have robust direction selectivity. Instead, it has some of the properties that MT lacks. It has orientation selectivity, color selectivity, texture selectivity. And so we think each of these areas is in fact specialized for a different type of signal, although we don't claim to know exactly what each area is doing. addition to being differently sensitive to different areas, I'll just mention also that we think these areas are processing signals in a hierarchical way. I mentioned that the signals first arrive in V1, which passes them to later stages. Those later stages pass further on to later stages. And as you get to later and later stages, what you see is the cells become increasingly more specialized to respond to particular types of stimuli. In V1, any cell will respond to an edge of the correct orientation of contrast, but basically they're just sensitive to whether there's a contrast border on their receptive field. As you get to later stages, you can find cells that become specifically responsive to complex texture patterns, and in fact at the very latest stages of the inferotemporal cortex processing of vision, you find cells that respond selectively to faces and only faces. So we think there's an elaboration of increasingly complex sorts of response properties. There's also division between areas that are much more involved in recognition and identification and representing what objects are, versus areas that are much more concerned about position, space, and the evolution of spatial positions, movements through the visual scene, such as you need to do visual navigation. So with this foundation, we wanted to explore how all this processing changes when you direct attention to different sorts of stimuli. And the type of attention I'm going to be talking about is obviously visual, but primarily visual spatial attention. In vision, you normally just look at what you're paying attention to. That's the most natural thing in the world. But you don't always. There are some situations, generally social situations, where you don't want everybody to know what you're paying attention to. And we cope with this perfectly well. We can keep our eyes directed straight ahead, but if there's reasons not to look at something, but you want to pay attention to it, it's easy to cast your attention to the left or to the right and pay more attention to stimuli in one location or another. So, for example, in a situation like this, Mr. Midshipman here is paying much more attention to the right half of his visual field than the left half of his visual field, knowing full well it won't be optimal if he moves his eyes to look at the object that he's paying attention to. If we took him to the lab, it would be easy to show that his performance on the attended visual field was superior. Sensitivity to small changes is greater. Reaction times would be faster. Thresholds for detecting stimuli would be better. These are the signatures of attention. These are what we call attention. In fact, that's the basis for our definition that I'm going to be using today for what we're calling visual spatial attention, the behavioral correlates of what happens when you attend to one location in the visual field. And the sort of rigorous design that we use for this is a task like this. This was developed in the 1980s by Michael Posner. Put a subject in a room, tell them to hold their gaze on a fixation point, and then very, very briefly flash faint targets, either to the left or to the right. Difficult to detect, they might see them half the time or so, but you can give a cue. You can tell the subject whether the target is more likely to appear on the left or on the right. And when you do that, what you see is performance is much better when the subject has a cue. And results like this are made up, but this is perfectly representative. If you look at performance detecting the stimulus as a function of its intensity, luminance, contrast, whatever you want to use, generally you get a psychometric function that looks rather sigmoid shaped. In a situation where the cue was valid, where the subject actually was told where the stimulus was likely to appear, you generally find that thresholds are lower. The curve is shifted to the left relative to the situation that would occur if, on a small fraction of the trials, you told the subject the stimulus would appear on one side, but it in fact appeared on the other. If the stimuli is very intense, attention doesn't matter. That's the bottom-up sort of thing. It'll grab your attention regardless. If it's invisible, attention can't save you. It won't pull things out of complete noise. But in this sort of intervening range, it's quite easy to get results like this, where you actually see performance going from maybe 75 to 25%, whether you measure it by detection ability or reaction time or so forth. This is the phenomenon that we want to look at. And it turns out, as a cognitive phenomenon, it's relatively easy to control and to measure. So unlike memory, which gets a little bit tough, or thinking, or other sort of higher processes, this is one where we really can look at it in a fairly precise and measured way. We want to look at what neurons are doing, so we can't do our experiments with undergraduates. We use rhesus monkeys instead. And rhesus monkeys are a great choice. They are extremely capable of learning these complex sorts of tasks that we require of them. Moreover, they come in with a built-in ability to hold their gaze fixed while attending to one side or the other. They live in tribes, and eye contact is actually a threatening movement on the part of a rhesus monkey. So they learn at a young age not to look directly at the alpha male if he's coming over looking angry, but they know how to direct their attention where they need to direct their attention. And so it actually makes it relatively straightforward for us to bring them into the lab and ask them to do tasks just like the one that I described for you. So we can train monkeys to hold their gaze on a fixation point. We monitor their eye position using a video system, and if they deviate, the trial simply ends and they have to wait for the next one to begin. While they're holding their gaze, we can present stimuli on either side and give them cues about whether a change is more likely to occur on the left or on the right, for example. And the cues we use vary. I don't think it matters how we cue them. It's really a matter of what they understand and how they're directing their attention. You can, for example, flash a small spot on one side before the trial begins. More often, we use blocks of trials. We start a block of trials with just one stimulus. The animal just works with that stimulus, and once he's got that going, we throw up the second stimulus. The animals learn to keep working with the stimulus that started there until we give another instruction trial in the beginning of the next block. That way we avoid actually having any sensory differences between the attend left and attend right situations. But as I said, I don't think the cueing makes a big difference, so I'm not going to focus on that very much. I'm not going to dwell on the monitoring of eye position. We worry about that a lot. But the fact of the matter is we have good control and know that the animal is in fact holding his gaze fixed on the screen. That's important for our experiments because what we're doing while they do this task is recording from cells in their cerebral cortex. And sometimes we use individual microelectrodes. More often we use arrays of about 100 microelectrodes that are permanently implanted into the cerebral cortex so that we can monitor many cells simultaneously. They remain extracellular. But we can isolate signals from individual cells based on the extracellular current flows, typically 100 microvolts or so, and monitor these action potentials and record activity from cells and their responses to different stimuli as the animals perform this task. And the first thing we do when we isolate a cell is arrange for one of the stimuli to fall inside its receptive field. So the cell is responding to one of the two stimuli on the screen, and then we can ask what happens to those those responses as the animal shifts his attention towards that stimulus or away from that stimulus. And these response histograms are quite typical of what we see. These are responses to many, many repetitions of the stimulus presentation, all from the same cell, both here and here. Same stimulus, same animal, same cell, same task. The only difference between these is that these trials were collected while the animal was paying attention to the stimulus in the receptive field, while these were responses to the same stimulus when the animal's attention was directed to the other visual hemifield. And this is sort of the common result that people find. It's not universal. Sometimes responses are weaker, but by far most cells will give a stronger response when the stimulus lies in the receptive field that we're recording from. The magnitude of the change that we get varies sort of systematically depending on what visual area you look at. I mentioned there's a hierarchy of visual areas in the cortex. Starting in V1, you tend to see relatively modest modulations there. Perhaps 5 or 10% is typical. As I'll show you later, it depends on how hard the animal is attending. So that actually isn't a veridical number. It just depends on the specifics of the task. But as you go to later stages, you typically see more and more modulation of responses. It's generally not all or none. It's not that the cells simply turn off when the animal isn't paying attention to the stimulus. It's typically a more modest sort of modulation. We're looking at percent here, so a 50% modulation is a pretty big modulation by the standards of cortex. So it's not the case that only the attended stimuli are represented in your brain. The whole scene is represented, but it's as if you sort of apply a filter that enhances some representations and not others. It's a fairly simple sort of adjustment, and it gets complicated in some situations, but in the simple case of one stimulus in the receptive field, with or without attention, it looks mostly like a gain change on the response. That is, it doesn't matter whether it's a preferred stimulus or a non-preferred stimulus, it mostly looks like you get a proportional change in the response from the cell. So for example, I mentioned cells in the middle temporal area are direction selective. They will typically respond best to one direction of motion, not at all to the opposite direction of motion, and have a sort of Gaussian tuning curve profile here. These are data showing the responses to the same stimuli, but with in red and without attention in blue. And you can see it didn't change the preferred direction or the sharpness of tuning, selectivity for different directions. It mostly just sort of turned up the gain of the response. The same applies if you look at orientation tuning in area V4 or contrast responses in area MT or other areas. Again, individual cells do a whole range of different things, but the typical response that you get from cells is just a stronger response. And so, more signal. Basically, these cells are providing more signals, or equivalently, you could say they're responding as if that stimulus was a bit more intense than it really is. That's important because it actually lets you discriminate stimuli a little better. For example, you could imagine it might be easier to tell that this direction is different from this direction than it is to tell those two directions apart with these responses shown down here. I will just mention, for that to really be true, we have to worry a little bit about signal to noise. The signal's stronger here. If the noise got much worse from the cells, then you wouldn't be doing any better. And I'll just mention that everything we've seen suggests the noise doesn't get any worse. In fact, it seems to get at least a little bit better. The noisiness of responses I'm referring to here is that if you take the same stimulus and present it over and over again, you won't get the same number of spikes from a cell. There's a lot of variance in how much response you get. And these data are quite representative. It's a raster plot just showing the responses of one visual neuron to many, many repetitions of the same stimulus. Everything kept as stable as we possibly could. But you can see on some trials we had lots of spikes, on others very few spikes for reasons we don't really understand. But this is what I'm calling the noisiness of the response. This affects your ability to tell what stimulus is in the environment based on what this cell is doing. And so the more noise you have here, the worse you're going to do in decoding what it's trying to tell you. Typically, you can look at these by just plotting the number of spikes you got per stimulus presentation, and you can see there's quite a bit of variance in here. If attention made that variance worse, we'd be in trouble, but the fact is attention actually makes that variance a little tiny bit better. It's not much to look at, but I'll make the assertion, whenever it's statistically significant, it's less noise, not more noise. And so this is just looking at the variance in the spike counts against the means of the spike counts with and without attention. There is very little difference here, but it's in the direction that supports the idea that attention makes neurons, sensory neurons, provide a little bit better signal to noise. The last thing I'll mention in terms of the phenomena that attention's been associated with is it also sort of affects the correlated activity of nearby neurons. If you have pairs of neurons that both respond to the same stimulus, they will both have some variance in their response. And when people have studied this, typically there's a very low level of correlation in their firing, so that when one is firing more spikes to a particular presentation, the other will tend to fire more as well. It's a weak correlation, typically on the order of a correlation coefficient of about 0.1, 0.1 or so. Not a very strong correlation, although it actually has real implications for how much information you can pull out of 100 or 1,000 neurons if they have that level of correlation. Attention does affect this correlation in a pretty substantial way. If you look at the correlation here called noise correlation, noise because we don't know where it comes from, using just a straight correlation coefficient, whether cells have moderate correlation or very weak correlation over here, attending to a stimulus reduces that correlation, and typically substantially. It'll cut it in half or so. We don't know why or how that happens, but that can have some real implications. It makes the cells a little more independent, which means that if you have a population of 1,000 cells that are more independent of one another, you can actually pull more information about the stimulus out. And the sort of way that people approach this kind of thinking is, if you had just two neurons and you wanted to decide whether it was one stimulus or another stimulus that had been presented, each stimulus will have some noise on every presentation, individual presentations represented by the dots. If you want to draw a line to try and distinguish one stimulus from the other, you'll make some mistakes. If they're correlated, those mistakes can go up in many situations, whereas if you decorrelate them, you can actually do a little bit better in distinguishing one stimulus from the other. And so these three phenomena, the stronger responses, lower individual variance, and lower pairwise correlation, are all thought to improve the quality of sensory representations. And we think this is probably a very big part of what goes on. And changes like this I've mentioned occur throughout the visual cortex, which lies here at the back of the brain. But they've also been seen in prefrontal cortex, where there are visual responses, in the thalamus, in the pulvinar, and in the brainstem, in the superior colliculus. And so what I want to do now is just turn to the question that I raised originally, which is, can we actually distinguish different components of attention? So, excuse me. I'm sorry. So most studies of attention of the sort that I've described have kind of considered attention to be a signal that sort of broadcasts throughout the brain, just one signal that turns up everywhere. And we wanted to see if we could subdivide it into distinct components. And the approach that we took was actually one based on signal detection theory. And I just want to take a minute to just familiarize you a little bit with the types of concepts I'm going to be talking about that are based on signal detection theory. Signal detection theory just basically addresses how easily you can distinguish a signal present from no signal present. And it all depends on the fact that you have noise in any sort of signal that you measure. If the target you're looking for is large compared to that noise, you'll have no trouble detecting when it's present and when it's not present. It gets much more interesting when the target you're looking for is relatively weak compared to the noise. And in that case, unless you have red and gray dots or different periods during your presentation, you're going to make mistakes. The best you can do is basically assign a criterion here that you'll use to say when you think there's a target present and when there's not, but you will make mistakes. And it's really these distributions that determine, in large part, your performance. In a situation like this, there are basically 4 outcomes that can happen. If the target's present, you could say it's present, that's a hit, or you can say it's not present, that's a miss. If it's not present, you can say correctly it's not present, or there can be a false alarm where you say the target's present when it isn't. Those behaviors are actually sort of determined by these distributions as a sort of construct. Here I'll turn them on their sides. So we have these 2 distributions, and we can draw a line and just say everything to the right now we'll call target present, everything to the left we'll say is target absent. And based on that, that will determine exactly how we do in terms of hits and misses, correct rejections, false alarms. And there are two specific parameters that are associated with the performance. One is how well separated these distributions are. Obviously, you'll do better if they're better separated, and that is the sensitivity measure, what I'll be calling D prime as we go forward. The other is where you draw the cut, the criterion for saying target's present or target's absent. And those 2 parameters basically capture your performance. You might think the criterion is preordained. You just put it halfway in between. That's often the case. If hitting a target correctly is as valuable as not reporting a false alarm, you'll probably want the criterion in the middle. But there are situations where detecting the target is much more valuable than the cost of having a false alarm. So, the guys screening our luggage at the airport really want to catch everything. Those targets are critically important. The false alarms are a pain for us, but that's okay. So in that situation, you put a much bigger premium on the hit than on the false alarms, and your criterion for what you would consider optimal performance would be in a slightly different position. But the main point I want to make is that these two measures basically capture everything. If you know the rate at which hits are being made, the rates at which false alarms are being made, or equivalently, just the rate of the misses and correct rejections, you can assign a criterion and sensitivity to the performance on a given task. And the very last thing I'll say is, you might think criterion is kind of a response thing, whether you will respond or not respond, the motor part of it, but that's not the case. You definitely have perceptual criteria. You see things as large or small, depending on their context. It's a decision you make about whether you perceive things one way or another. So we're talking about perceptual phenomena here. Why do we care about signal detection theory? Well, I had a graduate student in my lab, Thomas Lowe, who came and on his own just said, you know, we should try and split attention, and I've got a good idea. And he said, signal detection theory will work fine. We really didn't have any better approach. And he had made the observation that, in fact, a lot of the experiments that are done in attention, and in fact, all of the experiments that have been done on single unit recording in attention, in fact, might be explained by either changes in sensitivity, D prime, or criterion. And so he thought it might be a good way to approach the question about whether there are distinct mechanisms for those two parts of signal detection theory that might map onto attention. What he noted was that if you start at a given level of performance, say you're detecting half the targets, and you add attention so that you start to detect all the targets, or almost all the targets, you could do that either by shifting your criterion with no change in sensitivity, or you could do that by improving your sensitivity, your D prime, with no change in the criterion. So he went back and looked at some of the data we had from our monkeys, where we'd been asking them to attend to the left, attend to the right, and ask whether they were shifting their sensitivity or were shifting their criterion. And I think casually we had all been imagining that mostly they were shifting their sensitivity, more signal to noise, it sounds like sensitivity, that's what the monkeys would be doing. The neurons seem to be doing that. But in fact, when he looked at their behavior, he found that every animal he looked at, these are two of some of the animals he looked at, did both. And why not? They both help. So the animals became a little more sensitive. They could discriminate the stimuli a little bit better one from another, according to signal detection theory. But they also adjusted their criterion. They became more liberal about saying there's a target out there, because there was more likely to be a target in that location. And so both of these measures seem to be in play. This mattered for us, in fact, because we'd been just measuring changes associated with attention, which we hadn't really sort of considered in these terms before. And so these sorts of changes that I've described for you might be associated exclusively with changes in behavioral sensitivity, exclusively with changes in behavioral criterion, or some mix of the two, and we just didn't know. So the experiment to do seemed completely obvious. We just needed to train monkeys to direct attention by changing only their sensitivity or only their criterion, or do combinations on demand if we wanted them. The concept was very simple. The implementation took Thomas about a year to figure out how to get monkeys to direct their attention exclusively shifting sensitivity or exclusively shifting criterion. I will take you through just briefly so you get some idea about what's going on with these experiments, but this represents, I think, version 3 that he tried. On this task, it's very much like the task I described before. We briefly flash two stimuli, one on the left, one on the right. They disappear for a while, and then one stimulus appears. It might be on the left, it might be on the right. Depending on which side it is, the animal has to report whether it's the same or different orientation from what was there during the sample period. So, a little tricky, but pretty obvious. When you get your test stimulus, you have to say whether it's the same or whether it's different, but you don't know whether it's coming on the left or on the right. With this design, Thomas found that he could control sensitivity and criterion. And he could control it by just basically doing fine adjustments on the amount of reward that the animal got for different types of trials. To control sensitivity, what he did was to get a high sensitivity, he gave a lot of reward. To get a low sensitivity, he got a little reward, as you should expect. That's how it's going to work. But I told you animals will shift both sensitivity and criterion there. So to sort of titrate that out, he adjusted how much reward the animal got for a hit trial where there was an orientation change versus a correct rejection trial where there was no orientation change and the animal simply had to wait until the next stimulus appeared that would be a target. So by adjusting the rewards in 4 different ways, he could control sensitivity. On different days, he controlled criterion. That was a little more straightforward. The reward was always the same on both sides, but now it was adjusted to be higher or lower for hits or criteria, which shifted the animal's efforts for detecting targets or for detecting non-targets. And overall, this worked quite well. I won't go into a lot of detail here, but an easy way to show this is to plot the hit rate against the false alarm rate, which really captures the performance. On different days, in blue and on orange, he either controlled the sensitivity, trying to keep the criterion fixed, or controlled the criterion, trying to keep the sensitivity fixed. Each line joins 2 points where the animal went back and forth between high and low sensitivity in the receptive field location or high and low criterion in the receptive field location. We couldn't do sensitivity and criterion on the same days. The animals wouldn't handle that. That was just too much for them. So what we did was we picked one, went back and forth, tested for sensitivity, and then on a different day we went back and forth testing for criterion. This just replots the same data here as the difference, and it just shows that basically when we controlled criterion, we could get big changes in criterion with no change in sensitivity, and vice versa. I'll just point out, on both of these cases, the hit rate was going from 50 or 60 up to 80 or 90 or so, but in one case it was on the basis of a sensitivity change, and on the other days it was a basis of the criterion change. So once the animals were doing this, Thomas went into V4 and looked at how the cells responded there. We spend a lot of time looking at attention in V4. I should say it's not because we think that's where attention is. Attention is found in all of these different areas. V4 just happens to represent a really convenient area for us. It's relatively easy to get visual responses from lots of neurons. We are not in V1 where it's the easiest, but we're also not in the very latest stages where we have to find out whether it wants a face or a hand or something complicated. Basically, every cell in V4 will respond pretty well to those grading patterns that I've been showing you as I've been talking. You can get pretty good attention-related modulation. We could get more from the later stages, but it gets harder and harder to get the stimulus responses. We can get more responses from V1, but we don't get much attentional modulation. So it's a nice sort of crossover point for balancing those two effects. It's also easy for us to tell we're in V4 and not in V2 or later stages, and the receptive fields are good size. They don't span the whole visual field, so we can put a stimulus in and a stimulus out, and they're not tiny so that it's hard for us to get the stimulus into the receptive field. So just lots of convenient features. And to sum up, I guess, a couple of years of Thomas's life, this summarizes what he found when he went in and looked at responses from V4 neurons using this task. These are response histograms, basically plotting the period when the stimulus was on the screen, solid line for when the animal's attending to the receptive field, dashed line when the animal was attending elsewhere. And you could see we got a nice modulation here in the sessions where the animal was shifting his attention using sensitivity only. I'll point out here, we generally don't see much modulation in the very earliest parts of the responses. That's quite typical. I'll say we don't really know why that is, but that's a common observation. The main point from this slide, though, is on the days where the animal was shifting his attention using criterion, we didn't see any modulation in V4 at all. Now, the animal's hit rate was still going way up and down on those days, but there wasn't any modulation. And so based on this, we take away that V4 seems to be mostly involved in attention-related modulations associated with sensitivity and not with criterion. I mentioned that pairwise correlations in activity are also affected. We saw that as well. So on the days when the animal was shifting his sensitivity, we got a big drop as the sensitivity became greater, as we expected. But again, changes in criterion didn't seem to affect that much at all. So we assume now that V4 is mostly modulated based on changes in attention that are related to sensitivity changes and not criterion changes. But much bigger than that, we now think there's a good basis for saying whatever that criterion change is, it's a different component of attention. We can separate it out. We can see neurophysiological signatures here that are specifically related to one aspect of attention and not another. That obviously raises the question about what's this criterion effect coming from. And Thomas went on and did some recordings in the prefrontal cortex. There are visual responses up in this part of cortex. They are much more complicated in many ways. It's, as you might know, prefrontal cortex is associated with more like executive control, more sort of complicated rule-based representations, but there are visual responses there. In the animals that Thomas looked at, doing the attention task on visual stimuli, some of those cells gave visual responses, and many of them showed big modulations. And I should point out, these are very big modulations related to changes in sensitivity. These are the same changes in sensitivity of the sort that he was getting in V4, but here we get a much bigger modulation. Here, though, we found that there was also modulation related to criterion. Not as much, but in fact, in the same cells, we would find that there was changes in sensitivity and also changes in criterion from the same cells. So we think that V4, prefrontal cortex, are making quite distinct contributions to attention. There's no criterion signal we can pull out in V4. I don't yet know whether that's true of visual cortex generally, or whether criterion becomes a represented element as you get to the very latest stages of visual cortex, but I wouldn't be surprised if the visual cortex was exclusively related to the sensitivity component. We also don't know whether subcortical structures or thalamic structures have areas where you might find exclusively criterion sensitivity and not, criterion effects without effects on the sensitivity. So in the last part of the talk, I just want to describe some more recent experiments that we've been doing, which have been going on to look to see whether we can make a finer cut on what I've been calling sensitivity so far. Based on what I've told you, we think sensitivity and criterion are probably different components of attention. We're now no longer so foolish as to rule out the possibility that there might be other components that we haven't considered, or other cuts that might even be better and more sensibly a better description of what's going on in the brain. But what I want to talk about now is how sensitivity can be looked at in terms of both a selectivity element and also a sort of attentional effort. The effort part I can actually show you. So this is a sort of experiment that we asked the monkeys to do. You can try it yourself. You're supposed to hold your gaze on the spot in the middle. And in this case, there's no invalid cues. I'll just tell you, everything's changing on the right. What happens is after some random amount of time, the stimulus on the right changes its orientation by about 90 degrees. The stimuli turn off 2 seconds later. So if you see them go off without you seeing it, you missed it. But you shouldn't be missing this. This is 90 degrees. This is so easy, you hardly have to pay any attention at all. But I can fix that. Here's the same task, same stimuli, all the parameters the same, except now the change, which is still on the right, is just 15 degrees. Probably not so automatic. It depends where you're sitting, but we could tune this up if we had you in the lab. We could get whatever level of performance we needed from you. But with a stimulus like this, you should get the feeling like you need to do a little bit of work. You have to allocate your attention to that side. You should also feel like if the change had happened on the left, and there aren't any on the left, but if they were there, you might not detect them at all. So in the first version of the task, which was quite easy, you needed relatively little effort. In the second version, you needed more effort. And so we can describe that now by plotting basically your performance with the right stimulus against your performance with the left stimulus. And I'd say in experiments like I just showed you, all of the spatial experiments, spatial attention experiments I've shown you so far, we usually do this kind of thing where we ask the animal to pay attention to the stimulus on the right. Their performance goes up on the right, but goes down on the left. And then in another block of trial, we asked them to pay attention to the stimulus on the left. Their performance for that stimulus goes up. Their performance for the other stimulus goes down. And this sort of captures that notion of limited resources. You can allocate them for one side, but you'll withdraw them from the other, or vice versa. In terms of the effort that I asked you to allocate, what we're really doing is moving you along a line like this. If it's really easy, you don't have to put much more attention on one side than the other. In fact, if I flashed a strobe when the target came on, you could just stay right in the middle, keep your attention on both sides. You'd still get every trial right. So this is something you have under your control. You can decide how much effort you're going to put onto stimuli. You can put no effort on the stimuli if you want. So this lets us sort of think about this space of performance on the left and right in this way. So I've switched from Cartesian now to polar. And here I've done that because it kind of captures the elements I want to talk about. I've been talking mostly about sensitivity on one side or the other. But now let's talk about selectivity. You can have high selectivity for the right over the left, or for the left over the right, or equal selectivity for both of them. You're performing just as well on both sides. And the different eccentricities basically represent something like total effort, how much effort you're putting on. So you can pay equal attention to both sides and not very much to either side, or equal attention to both sides and lots to both sides. At some point, your resources do run out. You can't get to infinitely good performance. But within a local region, you can shift your behavior around like this. The fact is, no one had ever looked to see whether effort by itself without selectivity modulates neurons in the same way that our experiments did. We always sort of were just conflating the two, moving back and forth this way, where we were raising the effort on one side, but also the selectivity for that side. A postdoc in my lab, Supriyo Ghosh, did this experiment, basically trained monkeys where, again, using rewards, he could shift their performance around in this space of performance, now relative to the receptive field, inside the receptive field or outside the receptive field, from low effort in both places to high effort in both places, but keeping equal sensitivity in both locations. Now, in V4 and also now in the superior colliculus, he has found that both areas do show changes in activity, stronger responses when there's more effort in both locations. This shouldn't surprise you, because when there's more effort in both locations, there's more effort inside the receptive field. And so this could be driven entirely by just an increase in the effort in the receptive field location. But as he did this experiment, Supriyo was obviously filling out the whole plot here. He could move monkeys around this square from low effort both places to selecting outside, to high effort both places to selecting inside. And I won't go through the details, but it's all in the apple juice, how much apple juice you put in the different locations. Although, I will add, you can also move the animal's effort by making the task more or less difficult. If it's much more difficult on one side and easy on the other side, you'll get more attention to the side that's more difficult. And so he equally well could move the animals around just by changing the difficulty without changing the reward. The answer was the same in both cases. And what we had thought was maybe everything would be determined by how much sensitivity there was in the receptive field. So you'd have a high level when you were up here and a low level when you were down here. That turned out not to be the case. It's not as simple as it might have been. So shifting the amount of effort at a distant location, going between the blue and the green here, actually did affect the responses. Here as a bar plot, you can see. There was more response from the cells on the trials where the animal was putting more selection on the stimulus in the receptive field relative to when the animal was working well in both locations. In that case, the response was actually lower. So the cell was affected by the selectivity when we kept the sensitivity the same. And it's a little odd, too, because there's less activity from the cell when the animal's working harder, right? He's actually working harder, getting more trials correct overall in this situation than in this situation. So increasing the selectivity somehow increases the overall response. But these V4 cells are sensitive both to the overall sensitivity, the local effort the animal's putting into the receptor field location, plus how selective that is, whether he's excluding other areas as well. And so it looks like both selectivity and effort are components that will modulate the activity of V4. The very last thing I'd like to talk about is some sort of preliminary data we've done in one animal. I just want to mention it because it's really been our first foray into using optogenetic methods to manipulate activity in monkeys, and it worked. So given that effort, attentional effort, is something that looks like it's a potentially distinct component that should be considered in its own right, Supriyo wanted to go and look at the contribution of the locus ceruleus, which is a brainstem nucleus. You may have heard of it. It's largely implicated in arousal or attention, basically the amount of effort that's being put into tasks. And he wanted to see whether increasing the activity of neurons in the locus ceruleus, perturbing them with optogenetic techniques, could in fact affect the behavioral performance of animals. And so he used viruses to get locus ceruleus in one half of the brainstem to express channel rhodopsin so that he could shine the laser light on this nucleus and increase the activity during select intervals while the animal's performing the task. And so while the animal was performing the task on a random selected half of the trials, he activated the locus ceruleus in one hemisphere during the stimulus presentation, the sample stimulus presentation, while the animal was examining the orientation of that stimulus. And the last data slide I have to show you is that it worked. Basically, on trials when he stimulated the locus ceruleus, the animal performed better when the stimulus was on the contralateral side, where that locus ceruleus would have its effect. It wasn't a huge effect, but it actually shifted the performance up by, well, about a half D prime or so. It was a pretty good effect on that side. But it was selective for that side as well. When the stimulus was on the ipsilateral side of the stimulus, we got no effect. Potentially, the locus ceruleus then is playing a role in sort of controlling spatial allocation of effort for one hemisphere or the other, but maybe not. We don't actually know if the locus ceruleus ever naturally works in a push-pull kind of way. Most people think of it as kind of just one big nucleus that goes everywhere. Its projections are restricted to one side or the other side, depending on which brainstem half you're talking about. But conceivably, we've pushed the ceruleus into a mode of operation it doesn't usually use. We just need to get the physiology out on that, and I hope we'll be able to establish it soon. But to wrap up, what I'd like to just point out is I think the main points here, we think we've got an indication that attention, although we generally treat it as one thing, is probably made up of distinct components, as is memory, as is visual processing, as is virtually any system that you want to name. We think we can distinguish nicely using sensitivity and criterion between different components that exist in visual cortex or in other structures. It's not clear that's the best way to divide things up. It works for us. There may be superior ways to think about what the distinction should be. We don't think we've exhausted the number of distinctions. And in fact, the sensitivity part, we think, in fact, may be made up of components that would be usefully distinguished in terms of selectivity and the amount of effort that the animal puts into the task. But I should also remind you, for practical reasons, we've sort of stayed in a very local region here. It's conceivable that what's going on with different manifestations of attention might, in fact, involve other mechanisms as well. And in closing, I would just really like to acknowledge my collaborators on this, in particular Thomas, who did most of the work on the first part of the data that I described, and Supriyo, who's hard at work collecting data related to the effort component now. And thank you. Yes.
So you mentioned that if you change the difficulty on one side or the other, you would also see the selectivity or the effort change, so that like people would kind of like help on the one side that was harder. Is that right?
If you just simply ask the animal to perform better on one side. Yes, generally they will switch both their sensitivity and their criterion and their effort as well. Yes.
I thought you were mentioning that if you made one side, like if you're doing the same task but you make the right side harder, they would put more effort on the right side.
Yes. So in the last set of experiments that we described, sometimes we shifted the animal's attention left or right by simply rewarding more heavily for one side or the other. In other trials, we just kept the rewards fixed all day long for the left side and the right side, but made the task more challenging on the right versus the left, or vice versa. And that had the same effects in terms of changing the animal's performance on the left or right. When they're challenged more, they do better with stimuli on that side.
So did you notice if the change in how much effort they were putting from one side to the other was correlated to try to keep like a 50%, like trying to keep the same amount of performance for both sides, or they just like flat out perform better on the side that is harder?
They flat out perform worse on the side that was harder. So generally what we were doing was making the changes on one side big, like that 90-degree change, and the changes on the other side small, like that 15-degree change. They did worse on that side overall because it was harder there. What we did was probe performance with intermediate difficulty on both sides. So if this was 90 and this was 15, we'd probe with 30 here and probe with 30 here. They do much better on detecting the 30 here than over here. That was our proof that the animals were putting more attention, more behavioral effort on this side. So overall, they were doing worse on that side, but on the probe trials, they were doing much better.
Obviously very elegant work. I'm just curious, now, I don't have a feel for it. The last experiment, so on the contralateral side you get effect, and the ipsilateral side you don't. Is the interaction, can you pick up the interaction, that kind of stuff?
The interaction in the neurophysiology?
Yeah.
We haven't done that yet. So in principle, we can go back in and get all that neurophysiology while this optogenetic stimulation is going on. And I would expect we would see the same sort of thing, but we don't have the data. Question in the back.
I want to ask about the causal aspect of this. So if you were to microphone the V4 or the DLPFC, would you get shifts in sensitivity and your criterion?
Oh, that's a very good question. So without splitting things into sensitivity and criterion, people, not us, but other people have stimulated the prefrontal cortex, parietal cortex, superior colliculus, while animals do tasks like this. And what they find is the behavioral performance is better in the corresponding part of the visual field. They basically get more hits in that part of the visual field. And so on the basis of that, say it looks a lot like attention, maybe increasing this activity is equivalent in some way to producing attention. We haven't done those experiments, but it's actually a good question whether we would see selective changes in one type of behavior, sensitivity or criterion, by doing the stimulation. So we haven't done that yet, but it actually might be useful for that purpose.
So how universal is this, the interneuronal correlations going down with increased sensitivity or stimulus detection?
It's been seen now, I think, in every lab that's looked sees a change. It's now on the order of about 5 different labs have looked at it. The one outlier, which is interesting, is Marlene Cohen at the University of Pittsburgh had designed a task where, in fact, the correlation between two stimuli would, in fact, enhance performance, given the design of the task. And in that case, she saw the correlations go up for some of the cells. And so it's not always a downward change, it's sometimes an upward change. We have also seen that there's a bit of an interaction there with sensory processing, and maybe I could talk with you afterwards about some of the details there, because although the overall effect is a decrease in correlation, if you characterize particular cells with particular pairings of stimulus response properties, you can find some that go up reliably. But it's getting a little esoteric, and probably best for us to talk about that offline.
Yeah, I have a question, but I get to wait. So here.
Yeah.
Would you be able to go back to the third slide from Sabrio's experiment? It's the one showing the response times of the neurons. Yeah.
This one?
Yeah, this on the middle. So I found it a little like surprising that the timing of like the peak is pretty much the same in all the different conditions. Do you have any like intuition, or is that kind of typical?
You mean that all of them have the same structure, in particular a sharp peak here and a secondary peak?
Yeah, most notably the very first peak in the back end. It's all at the same point in time.
The timing doesn't surprise me so much. It's the same stimulus coming on in the receptive field. So attention, we've looked carefully at whether attention affects the latency of neuronal responses. After all, changing the contrast or intensity of a stimulus affects the latency a lot. Attention doesn't affect the latency of responses very much at all. There's a very minor effect. So I'm not surprised that the peaks are all in the same place. I'm also now used to the fact that attention generally doesn't make much difference in the activity peak in the earliest part of the response.
But doesn't attention not modulate the reaction times and response times?
It does. The behavioral reaction time and the behavioral response times, which would be coming, for most tasks with monkeys, not in this task. There are no response in any portion here, but if you ask the animal to respond as quickly as he wanted, typically 250 or so, something like this, the animals would be beginning to respond. The distribution would sort of go out over 100 milliseconds or so. So even though the onset of the neuronal responses in sensory cortex isn't faster, it doesn't mean that you can't pick up speed in later stages of processing.
So let's take two more, and then we'll, from our, here, and then we'll take about a half-hour break, and the next talk will start at 10:45, but of course, you can all sort of come down and talk to Dr. Maunsell some more while that's going on. But so we'll take Professor Dagger first.
Thanks. That's really interesting. I think I understand selectivity and sensitivity, but I'm not sure I've gotten how criterion works. Could you give an example of how criterion works with a richer kind of stimulus?
A richer kind of stimulus.
Something like this. So suddenly I put this up and your attention shifts to my water bottle.
So the criterion we're using is this your target or is this not your target? So in all these experiments, basically the animal's being asked, is this the target that's different than the stimulus that you saw before, or is this not the target from the stimulus you saw before? And I should say, sensitivity is a bit simpler than criterion. There are multiple types of criteria in play in any task. So we were quite explicitly looking at the target, non-target criterion. You could also, if you wanted, for example, have come up with a criterion that said, am I going to be responsive and say target on the left versus target on the right? And some of our tasks preclude that, but others don't. And that's a different criterion operating in parallel, but that's not one that we look at in terms of being able to distinguish what's going on. So this was really, do you see a target here? Do you not see a target here perceptually?
Which is connected with the kind of reward that the monkey can be expecting.
Yes, definitely. The animals know which side will generate what rewards for a target and what rewards for a non-target, and that is the basis on which we adjust their criterion. So the target becomes much more valuable than the non-target.
Do you have any intuition as to why that initial spike doesn't change in amplitude? Because that kind of runs counter to that simple story that attention modulates neuronal gain, doesn't it?
It does. And I don't have any good model I can give you or provide for you in that way. I will just say, although it's not visible on this plot, well, sort of is, the animal has his attention allocated throughout the whole trial before the stimulus comes on. And in fact, you see in some cases, and I'm not going to try and defend this one, you actually see a modulation before the stimulus comes on, after the stimulus goes off. And if you get rid of this transient by making the stimulus a little weaker or ramping it more slowly, you just see the modulation throughout the whole response. This is not a delay in turning on the attention. It's there the whole time, before, during, and after. So we think it's probably more related to gain control adjustment in the local circuit, that somehow when you suddenly ramp up to a point where the gain control has to bring it down a lot, that somehow just swamps what's going on. But it's all hand wavy. I don't have a good explanation for you, but it is, there's no reason to think that's the latency of attention turning on. Attention is there. It's just something swamps it during a transient. If we get rid of the transient, you'll see it in the earliest parts of the response, no problem.
All right, so we'll take our half-hour break and then hope to see you all back in maybe even